Welcome to the basics of neuroscience. Today, we are exploring a truly miraculous event, neural tube induction. Imagine trying to build a massive, complex skyscraper, which represents the human brain and spinal cord. You cannot just start throwing bricks together. You first need a master blueprint and a solid foundation. In this lesson, we will discover exactly how a simple flat layer of cells receives a chemical wake-up call, instructing it to fold into the magnificent structure that will eventually become your entire central nervous system. Let us dive in. Now that we know what we are building, let us look at the starting materials. In a developing embryo, the body begins as a simple, flat stack of 3 pancake-like cell layers. At this very early stage, the top layer, called the ectoderm, does not actually know it is supposed to become the nervous system. It is just sitting there, waiting for instructions. It needs a specific wake-up call, essentially a chemical text message, from the cells located right beneath it. This crucial wake-up call, which tells that flat sheet of cells to change its identity and eventually roll up into a tube, is exactly what we call neural tube induction. Understanding that the ectoderm needs a signal leads us to ask, where does this signal come from? During the 3rd week of human development, a rod-like structure called the notochord forms in the middle layer, which is known as the mesoderm. You can think of this notochord as the primary signaling center, or the director of the embryo. It begins secreting specific chemical molecules called morphogens, such as sonic hedgehog. These morphogens travel upward from the notochord to the overlying ectoderm. When they arrive, they instruct those naive top-layer cells to change their identity, transforming them into specialized neuroectoderm cells that are now destined to become nervous tissue. So the cells have received their chemical text message, but what happens next? Once the ectoderm cells transform into specialized neuroectoderm, this newly specialized region begins to physically thicken. We call this thickened area the neural plate. In a beautiful mechanical process, this flat plate begins to fold inward upon itself, eventually fusing at the top to create a hollow cylinder. This cylinder is the neural tube. Ultimately, the front part of this tube will balloon out and expand to become the complex structures of the brain, while the rest of the long tube will become the spinal cord. This folding is the physical birth of the central nervous system. Before we look at the mathematics of these signals, we must understand why this matters in the real world. Understanding neural tube induction is absolutely critical because it is the very first step in creating the human mind and body control center. If this delicate process fails or is interrupted, the neural tube does not close properly. This leads to severe, often fatal birth defects known as neural tube defects. Conditions like spina bifida, where the spinal cord is exposed, or anencephaly, where major parts of the brain fail to develop, are direct results of errors in this exact process. This is precisely why pregnant women are strongly advised to take folic acid supplements, which help ensure these cellular signals and folding mechanisms work flawlessly. Now that we understand the biological importance of these signals, let us look at how they actually spread out physically. The notochord releases a signaling protein that diffuses away from the source, creating a concentration gradient. We can model this using an exponential decay formula. In this equation, C of x represents the concentration of the morphogen signal at a specific distance x from the notochord. C0 represents the initial maximum concentration right at the source. The constant e is the base of the natural logarithm. Finally, lambda represents the decay length, which measures how far the molecules can travel before being absorbed. When the distance x increases, the concentration drops exponentially, meaning cells further away receive a much weaker signal. Let us see this formula in action with a concrete example. Imagine a developing embryo where the notochord secretes a signaling molecule with an initial concentration of 1,000 molecules per cubic micrometer. The decay length is 50 micrometers. We want to find the concentration at a distance of 100 micrometers away. We simply plug these values into our exponential decay equation. The exponent becomes -100 divided by 50, which simplifies neatly to -2. The value of e to the power of -2 is approximately 0.135. Multiplying this by our initial 1,000 gives us exactly 135. Physically, this means cells at this location receive a significantly diluted signal, which might instruct them to become a completely different type of neuron than those closer to the source. Now that we have used the formula to find concentration, let us tackle a classic threshold problem in developmental neurobiology. In the developing neural tube, motor neurons will only form if the signal concentration is at least 50 molecules per cubic micrometer. If the initial concentration is 400 and the decay length is 40, what is the maximum distance where motor neurons can still develop? We need to find the exact physical boundary, the distance x. We start by setting up our equation with all the known numerical values. Our primary goal here is to isolate the variable x, which is currently trapped inside the exponent on the right side. This setup establishes a clear path to algebraically solve for the boundary line. Continuing with our derivation, we must first isolate the exponential term. We divide both sides of the equation by 400. The fraction 50 over 400 simplifies neatly to 0.125, or 1/8. This leaves the base e and its exponent completely alone. To bring the variable x down from the exponent, we take the natural logarithm of both sides. The natural logarithm of 0.125 evaluates to approximately -2.079. By doing this, the natural log effectively cancels out the base e on the right side, leaving us with a much simpler algebraic expression to work with. We are now at the final stages of our derivation. We have a simple linear equation. The negative signs on both sides of the equation will cancel each other out, making everything positive. We simply multiply the value 2.079 by the decay length of 40 to isolate x. This final arithmetic step gives us 83.16. The maximum distance at which motor neurons can form is approximately 83.16 micrometers from the notochord. Physically, this creates a distinct functional stripe of motor neurons in the developing spinal cord. Any cell located further away will not receive enough signal and will develop into a different type of cell. This mathematical boundary perfectly explains how the spinal cord organizes itself. Having proven the mathematical boundary, let us visualize this entire process. On screen, you see a two-part diagram. The top half shows the physical anatomy, the notochord sitting in the mesoderm, sending inductive signal arrows upward to the ectoderm, causing it to thicken into the neural plate. Directly below this, we see the mathematical reality. The graph shows morphogen concentration on the vertical axis and distance on the horizontal axis. Notice how the concentration curve starts at its highest point, exactly at x equals 0, right at the notochord, and then drops off exponentially as you move away. The dashed horizontal line represents the cell fate threshold we just calculated. Where the curve drops below this dashed line, the cells no longer receive enough signal to become motor neurons, beautifully illustrating how biology and mathematics map out the nervous system. We have covered a tremendous amount of ground today, so let us summarize the key takeaways. First, neural tube induction is the miraculous starting point of the central nervous system, transforming a flat layer of cells into a complex tube. Second, this process is driven by the notochord, which secretes chemical morphogens to instruct the naive ectoderm cells. Third, we learned that these signals do not spread randomly. They follow a strict exponential decay gradient. Finally, by understanding this mathematical decay, we can predict the exact physical thresholds that dictate cell fate, mapping out the highly organized layers of the brain and spinal cord. You now understand both the biology and the math behind how your nervous system was built. Excellent work.